Hi, I'm Lee Kelso, host of the HealthCall Live Radio Hour. I'm glad you're here as we learn more about a chewing gum that may be able to block the transmission of the COVID virus. It is in development right now at the University of Pennsylvania School of Dental Medicine by Dr. Henry Daniell. Thanks for joining us, Doctor.
Thank you very much, Lee. Appreciate it.
So let's get to the basics here. This chewing gum blocks the transmission of the virus. It does not treat the disease, correct?
No, it actually blocks reinfection. So what it contains is a standard cinnamon gum, but in addition, it contains a protein. It's called a virus trap protein, but this protein is naturally produced in the saliva and it's also present in the blood. But it's not in sufficient amounts. If we have been producing more of this protein in our saliva, it could essentially bind the virus and block it from entering. So the way it works is there are two approaches. One is this protein called ACE2. It directly binds to the virus spike protein and traps the virus in the chewing gum. But if it does not trap, then the virus obviously will try to enter the human cells through the oral epithelium, and there it needs to get into a gate, and that is the ACE2 receptor. And if this enzyme binds it, then it closes the gate. So it prevents reinfection. So step one is it protects the patient because all the COVID-19 symptoms are proportional to the viral load. And this is why some people don't go to the hospital and others go because the virus replicates more than others. So the viral load, when it is decreased, it gives COVID-19 at the time of early infection to fight back to protect themselves. And our studies show that when we put even small quantities, 50 milligram, the gum is 2 grams. If we put a 50 milligram, it still gets 95, 97% blockage of trapping. And we also showed in the publication that it blocks the gate, it blocks the entry as well.
So I'm reading that as this gum sort of acts like flypaper to grab onto the virus and get it before it can infect your cells. Is that the simple version of what you just explained to us there?
Excellent analogy. Excellent analogy. It's like a flypaper, except that the flies that don't stick onto the paper, right, still can get away. In this case, it doesn't because the enzyme goes and binds to the same gate as the virus, and therefore it closes the gate. So it traps like a flypaper, but at the same time, if it doesn't, then it goes and blocks the entry as well.
Gotcha. So dual action, right?
Right, and this is why when people vaccinated people also get breakthrough infections, unvaccinated people, early infection period, they don't have symptoms, they don't know, so they still continue to spread the virus. And this one essentially works like a PPE, except that it also helps the patient. The current PPE is protecting others, but in this case, this PPE actually protects the patient first.
So is it your vision that I would chew this gum frequently during an outbreak to prevent infection?
Exactly. The first step is the decreasing the viral load, and so for that to accomplish for this particular pandemic, for the SARS-CoV-2, we have used the enzyme that's naturally present in the body. But for the influenza and various other viruses, we are also developing that's a different trap protein that we don't have. And so we put a different protein. So in principle, then, yes, it varies from virus to virus. And in this case, the spike protein is present in all variants. So this particular one doesn't matter. It is very highly efficient in trapping different variants. Right now, we are working with the Delta variant, and we are waiting for samples for the Omicron as well.
Gotcha. So it may be that we would see products available for the flu, for COVID, for a number of different viruses that are in circulation.
Correct. Before this publication, in our Penn news release, they also showed another publication where we used the chewing gum with enzymes that break down the plaque and kill the bacteria that causes tooth decay. So there our protein was killing the bacteria and softening the plaque and getting through it to kill the bacteria that adheres very close to the tooth. So yes, so this is a platform technology which actually helps in killing, neutralizing viruses or bacteria in the oral cavity.
Very interesting. So where are you in development? I understand you're hoping for an emergency use authorization soon, but give me the timeline. What are you expecting here?
Right. So all of our studies that's published so far is using clinical samples. We have taken the patient sample, saliva, we put the gum powder, and then we looked at how efficient it is in trapping the virus. What we don't know, which after we get the emergency use authorization, we will learn, is how long does it take for the virus to repopulate in the saliva. Our current estimate is about 4 hours or longer. If the virus repopulates in 4 hours, then they may have to take a second gum at that time to continuously keep the viral load lower. That is for long-term therapeutic purposes. But for short-term, right now, my lab is in the dental dental clinic, where the patients come and sit in the patient chairs and open their mouth wide. So during that period, for 1 hour or longer, this decreasing the viral load protects the healthcare providers and transmission, because when you use aerosol generating procedures, that is a big problem, or when people are in public transportation and so on. So short duration is already done, so we know, but longer duration, how frequently the gum needs to be taken, that we can get the data only after we get the emergency use authorization.
And what's your estimate? Do you see this going to market before the pandemic converts into becoming endemic and the virus is just everywhere and it's no longer an urgent crisis?
We have used 2 production facilities in our publication, and so we have already shown that these facilities are capable of making it and they're ready to go. So we could make few 100 million chewing gums in the next 3 months or so. And so the other big advantage in this is that this protein, ACE2, and other proteins in the chewing gum are stable up to 3 years when stored at ambient temperature. So large majority of the globe is still not vaccinated, and so they are still waiting for affordable ways without cold chain, without refrigeration, and therefore this is very helpful at this current stage of pandemic, because the decrease in transmission, decrease in viral load helps the globe, no matter where the outbreak happens. And so, yeah, we are ready to go. As soon as we get the emergency use authorization, we are ready to scale up and launch this.
So I want to get to the moment when you're in the development process. You're working with the ACE2 receptor, I understand. That was kind of the focus of your research. You hear that COVID attaches to ACE2. Give me that moment when the light bulb went off, and how did that thought come to you?
Very, very smart question. Yeah, we have been working with ACE2 for many years. So this is not the first time we started working with the ACE2. What happened is when the this outbreak, COVID-19 outbreak happened, that immediately it was clear that the virus binds to ACE2 and enters the human cells using the ACE2 receptor. As soon as that information became available, I immediately thought that, well, if it binds to the receptor, it should bind to the enzyme also. So if we make the enzyme, then it should outcompete the virus for the receptor. But what was a surprise was that the virus also binds to the enzyme and inactivates it. In fact, a lot of these diseases are called, in scientific terminology, it's called the RAS pathway. This enzyme is a key enzyme for a lot of metabolic disorders. In fact, we were working with hypertension and heart failure because limitation of this enzyme is a fundamental reason for those symptoms and diseases. So we were developing this ACE2 to treat hypertension and heart failure, and we are still working on that. But then we switched this to make a gum so that we can control the virus at the point of entry.
That must have been a very exciting moment for a scientist like you to see that potential and say, "Hey, this could really work." I'd like to hear a little more about how that felt and where you were when you had that awareness, and your sense of what did you feel when you saw those first lab results and saw how effectively it was trapping the virus?
Yeah, and this is one of the wonderful things about science. When we plan something and then things work as expected, it's quite exciting that our hypothesis works. But in this case, nobody showed before or even suggested that the enzyme and virus directly interact. If there are 1,000 papers on ACE2 receptor, there was nothing on the enzyme. So but scientifically it would say that the both should interact because that's the same mechanism that which this enzyme goes through its own receptor. And therefore, yeah, and especially it was mind-boggling when we had this cell phone camera technology, which was also developed here at Penn. We could see this virus as bubbles. And each virus forms a bubble when we add a chemical to that, and we could take the pictures in a cell phone camera. When we looked at the placebo gum and the gum with the ACE2, the bubbles disappeared. And this was, of course, was predicted by me, but it was so wonderful to see that. And then we didn't stop there because, no line of research will survive with a single experimentation or line of evidence. So we started multiple lines of research. Then we had a pseudovirus with the spike protein, which will enter the human cells. Then we added this enzyme to see whether it is going to block the entry. So we did several pseudovirus experiments, and we also did protein, we did PCR, and a lot of other lines, and everything converged to the same thing. So the real eureka moment was that, well, multiple lines of evidence showed that this is working. So that's when I felt quite excited and relieved at the same time.
That must have been an exciting moment. I congratulate you on experiencing that. Has anything like that happened in your scientific career before today?
Yes, we have had this. Years ago, we developed the anthrax vaccine, and we immunized the animals and shipped it to NIH. And at 3 a.m. I had an email saying that all the animals survived the anthrax challenge, the anthrax spore challenge. And I think science, it's wonderful when things work. And we have had other moments like this before.
So just one final little bit here. So the process is you are infusing a lettuce plant, I understand, correct? That's where this enzyme is coming from. You are creating it in form of lettuce?
Correct, correct. And I could send you some images of how we create these plants. And it's a pretty simple, straightforward process that we have used in our lab for over 25 years. We clone this gene, human gene, and we have what's called as a gene gun. And we shoot the gene using a gene gun into plant cells. And once the gene gets integrated into the plant genome, forever and ever and ever, it will produce this protein. It will consider this protein as its own protein and keep producing it. And then we have a commercial facility here where we give the seeds of these plants to this facility where they are sown by robots and they are grown under FDA approval conditions. And then the leaves are freeze dried. FDA expects us to measure the bioburden, any microbial contamination, and also moisture content. And then we do the drug dosage and use that plant powder, freeze-dried plant powder. We ship it to the chewing gum company to make the gum.
So, Doctor, if you were to guesstimate when this might come to market, is there a timeline you have in mind?
If we get FDA emergency use authorization, or not, in fact, we have gone through one round of review and we have already answered the questions. Answered means they want more toxicology studies to be done, and we have finished all of that, and we are now resubmitting that. And once we hear back from them, the timeline would be 2 to 3 months, where we could, we already have gum which we can test in the clinic right now. And this is to find out how long it takes for the virus to repopulate. And some of those will be done as soon as we get it. But large scale production with 100 million gums and so on will take 2 to 3 months.
I'll look forward to watching your progress, and I hope to see that product on the market soon. Dr. Henry Daniell, thank you. Appreciate it. Congratulations. Great to see you.
Thank you so much. I really appreciate it. You know, I also want to add that you are playing, this is an entire village, and you are also playing a very important role in this because at this stage, until now, I write grants from NIH, I make things happen, innovation side. But beyond this, going to the commercial scale production, investors get to know about science. Investors don't read scientific articles, and interviews like this are enormously helpful in moving the product from innovation to the end users. And therefore, I want to thank you for doing an in-depth interview, because the questions that you asked and the responses that I gave are going to be very important for people to evaluate and advance the technology.
Well, thank you. I appreciate that. That is Dr. Henry Daniell. He is the Vice Chair in Department of Basic and Translational Science at the School of Dental Medicine at the University of Pennsylvania. Thank you, Doctor.
Thank you so much. Bye-bye.